Okay, good morning. On behalf of the University of Wisconsin and Qualis Transporters, I'd like to welcome you to today's webinar, Wholesale Models Integrating Transport, Metabolism, and Regulation to Predict Cholestatic Hepatotoxicity. I'm your host, Eric Buxton. Today's speaker is Dr. Kenneth Brower, who is Associate Dean for Research and Graduate Education at the UNC Eshelman School of Pharmacy. Dr. Brower has over 25 years' experience in preclinical drug development and is co-founder of Qualis, a provider of hepatic drug transporter products and research services. Prior to founding Qualis, Dr. Brower served as Executive Director, Drug Metabolism and Pharmacokinetics at PPD Discovery, where he directed research in metabolism, pharmacokinetics, toxicology, and absorption technologies. Previously, he served as Director of Preclinical Development of Drug Metabolism and Pharmacokinetics at GlaxoSmithKline, and was responsible for drug metabolism and pharmacokinetic strategy leading to candidate selection, review of candidate project plans prior to candidate selection, and developing and implementing processes to ensure a smooth transition from candidate selection to full development. Dr. Brower has over 25 publications in peer-reviewed journals, holds 2 patents, serves on the editorial advisory board for the Journal of Pharmaceutical Sciences, and is adjunct faculty at the Division of Drug Delivery and Disposition at the School of Pharmacy, University of North Carolina. Following today's presentation, there will be a time for questions and answers, and also the slides for today's presentation will be sent out to you shortly after the talk is complete. Dr. Brower?
Thank you, Eric. I'm going to talk to you today about work that we've been doing to develop a wholesale model that integrates transport function, metabolism, and regulation in order to better predict cholestatic hepatotoxicity. Some of the things that you need to take into consideration when you're trying to predict this, especially from in vitro systems, is that you must have transporters and metabolic systems for both the test compounds and for bile acids. In addition, you want to have a system that generates in vivo relevant intracellular concentrations, because this is what's going to drive many of the interactions that occur inside the hepatocyte. And this intracellular concentration, as we know, is a function both of a hepatic uptake and efflux, including basolateral and canalicular efflux in the hepatocyte. And also metabolism can play a role in this also. From our last seminar a couple weeks ago, we've shown that the adaptive response by the liver is a very important component to consider when you try and predict the potential of a compound for cholestatic hepatotoxicity. And the interplay of these systems is also very important in order to be able to predict. And the sum of all these things, we have concluded that it really requires a wholesale system in order to integrate all of these parameters. And so it's especially important for transporters to have a polarized system. We've shown before that plated hepatocytes on a single layer of collagen is not a polarized system. You do not have bile pocket formation, and the canalicular efflux transporters especially are not functioning, but you do have good uptake transporter function, which is why we've been working with a sandwich cultured model. It is one of the systems where cell polarity is reestablished. You have uptake and efflux transporter function. This includes basolateral and canalicular. It's a metabolically competent system. And as we have shown in the last talk, the regulatory pathways, especially for bile acids, are intact and functioning. It's also a system where you can manipulate the culture media conditions, i.e. with the addition of protein, in order to generate in vivo relevant unbound intracellular concentration. So it allows you to do a lot. What we have been focusing on is cholestatic hepatotoxicity, and in particular hepatocellular injury. There's many types of toxicity in the liver, including bile duct injury, and there's also a mixed pattern of that it can include factors from both of these types of injury. But for our purposes, since we are dealing exclusively with hepatocytes, we're focusing on hepatocellular injury for that. So in the past, in order to predict cholestatic drug-induced liver injury, the historical hypothesis has been that if you inhibit BSEPT, you'll get higher intracellular concentrations of bile acids. These are detergents which dissolve membranes, and at high intracellular concentrations will lead to hepatotoxicity. However, in recent years, people have recognized that these bile acids can also cause toxicity through various types of signaling events, including some death receptor activation. And we've shown that drugs can cause increased bile acid levels by directly inhibiting various transporters, both on the canalicular side, but also on the basolateral side. And another component that we've shown is important is blocking the cell's response to higher intracellular concentrations of bile acids. And an example of this would be FXR activation. And it's very important to remember that the liver has a very high ability to compensate. We call this the adaptive response, and that BSEPT inhibition alone we do not think is enough to cause cholestatic drug-induced liver injury. The liver can adapt to a certain extent to that. And this is shown if you look at some of the work that's been published previously by Dawson and also Morgan, but you can see that there's a very weak relationship between the potential of a compound to inhibit BSEPT, and this is human BSEPT IC50s measured in vesicles for this. There's a very weak relationship between that. There's a number of false negatives and a number of false positives with that. And so based on some of this data, we decided to go and look and see what other factors may be important in this. And as we shown last time, the adaptive response we think is very important, and this occurs relatively rapidly. So in sandwich cultured human hepatocytes, if we expose them to cyclosporine for various time periods here, and we're looking at the biliary excretion of an endogenous bile acid, we can see that administration of a cyclosporine A, a potent BSEPT inhibitor, rapidly decreases the biliary excretion of endogenous bile acids. In the same system, at the same time, if we look at what's going on inside the cell, we can see as we're inhibiting efflux of bile acids, the intracellular concentration of bile acids is increasing over time. And at some point, it gets high enough where we begin to activate FXR. And this will lead to suppression of bile acid synthesis, and also, as we showed last time, induction of basolateral efflux transport for this. So what we really needed to come up with is something that can integrate these multiple mechanisms. And this cartoon demonstrates kind of our train of thought for that. So bile acids shown in green here are taken up by NTCP and other transporters and effluxed into the bile through BSEPT. If we then have some initiating insult, and in this case I'm showing inhibition of BSEP. So now we've blocked the efflux of bile acids from the hepatocyte into the bile. The intracellular concentration of these bile acids will continue to go up because it doesn't have one of the major pathways of efflux has been blocked. And at some point, those intracellular concentrations of bile acids will get high enough to activate FXR. And as we said last time, activation of FXR will allow the hepatocyte to respond by increasing basolateral efflux transporters, primarily OST alpha and beta. And this will then, this additional clearance pathway that comes on board from FXR activation will then drive down the intracellular concentration, or you'll reach some new steady state level for that. So the cell, or the hepatocyte in particular, is capable of adapting to higher intracellular concentrations of bile acids. However, if you have an additional insult, say FXR antagonism, so you block the cell's ability to respond to increased intracellular concentrations of bile acids, or you inhibit the basolateral efflux pathways, now you've blocked the ability of the cell to respond to increased intracellular concentrations, and you've blocked all of the efflux pathways for that. And what happens then is then the intracellular concentrations of bile acids will continue to build up, and you'll see toxicity for that. So compounds can increase the intracellular concentrations of bile acids, as we said, through BSEP inhibition or inhibition of basolateral efflux transport or FXR antagonism. So there's multiple mechanisms that can occur to generate higher intracellular concentrations of bile acids. So what we wanted then to look at is, well, what parameters, if we really wanted to estimate cholestatic drug-induced liver injury potential, what parameters would we have to look at? So one of the things you would want to look at would be IC50 values for both the parent and relevant metabolites against BSEP and other transporters that were involved in the hepatobiliary disposition of bile acids. You would also want to determine an IC50 value for the potential of your compound to cause FXR antagonism. So you would have to get IC50 values for all of those, for parent and metabolite for that. And then in addition, there may be other parameters that you would want to consider, again, both for the parent compound and metabolites for this. So you'd want to know Ki values for the test compound for transporters that are relevant to determine the uptake and efflux of your test compound, because that's going to drive the intracellular concentration. You may want information on protein binding, both outside the cell and inside the cell. Some idea of the intracellular concentration so that you can relate these IC50 values determined in perhaps a vesicle system. So you can relate those IC50 values to the actual intracellular concentration of your test compound. You need to consider, as I mentioned, the impact of metabolism, because that will obviously change the intracellular concentration of the parent compound. And you'll need some type of modeling or algorithm to try and integrate all of those together. And when we thought about all of those, that's an awful lot of information to have relatively early in drug development for a screen. In fact, the cost of obtaining all those parameters is prohibited, and there's many parameters there that are very difficult to determine early or even later in development. So this led us to understand that you really needed an integrative assay to have some potential to predict this early in drug discovery or early drug development. And so we did look in the literature for examples of where people have tried to take this individual approach. So here's an example where the test compound they had characterized for IC50s against various transporters, and you can see here it was moderately potent for human BSEP. For MRP2 they actually saw a stimulation, fairly potent against MRP4, IC50 of around 6, and it was also a fairly potent FXR antagonist. And they also, in this example, had some preclinical toxicity data where they did flag it for hepatocellular necrosis. However, this compound was indomethacin, which is a fairly safe drug. So just emphasizing the difficulty that you can have trying to integrate some of these individual parameters. And so what we tried to do is put together an assay that would do this for us. And we wanted a mechanism-based assay. We wanted a whole cell model. It had to use human hepatocytes. There's a very significant amount of data out there indicating species differences in bile acid synthesis, metabolism, and regulation between species. So we wanted to focus this all on human. And the other thing that we were interested in, we wanted the readout to be fairly simple and that would reflect bile acid induced toxicity for this. And of course, we wanted it to be predictive of cholestatic drug-induced liver injury. We initially identified a test set of 50 drugs, and I will go into those in more detail very shortly. So looking back at all the pathways that are involved in cholestasis, and this is an example of an adverse outcome pathway for cholestasis from a publication by Finken in 2013. And when you look at this, it's kind of obvious you're picking up something very early here and trying to predict something later. And there's a lot that happens in between here that you're just totally ignoring. And this is really, everything in here is the hepatocyte trying to respond and adapt to these higher intracellular concentrations of bile acids. So one of the things that we wanted to do was try and integrate all of these together in a single assay. When you look at just the predictive power of BSEP inhibition alone, it's not very significant. You can see there's a couple of them that you get right based on the potency for BSEP, but there's a lot of false positives here and a lot of false negatives. And in general, it has very low accuracy. So BSEP, I think the take-home message here is using BSEP inhibition alone is not going to get you very far down this pathway of being able to predict cholestasis for this. So what we've tried to do is to take this same scheme and look at BSEPT inhibition, but also integrate all of the adaptive responses and the regulatory responses that the cell has in order to be able to better predict cholestatic hepatotoxicity. And so the assay that I'm going to talk to you about integrates all of these pathways. And so it's a 96-well format. Again, it uses human hepatocytes. And what we've done is optimize the culture conditions for various factors. One, we culture these for 5 days, so we have optimal formation of bile pockets and efflux transporter function. It takes time for these hepatocytes, once they're plated, to repolarize and have good efflux transporter function. We also identified and developed a special media which we call the sensitization media, and I'll talk more about this in a minute. But basically, it creates an environment using lipids and bile acids that allows us to see an effect in a relatively short amount of time. We can also, and typically do, run these studies in a standard culture media, which lets us account for direct toxicity of your compound. And as I said, we wanted a relatively short experimental period, so it's a 24-hour incubation, and this allows enough time to integrate the inhibition of transporters, it allows metabolism to occur, and it allows the regulatory component, or the adaptive response time, to take place. And we have a single readout, which in this case is an LDH measurement for this. As I mentioned, we spent a lot of time optimizing the assay conditions in order to test for disruption of bile acid homeostasis. And the sensitization media is it's a cell culture media that creates the conditions with physiologic concentrations of both bile acids and lipids, which allow hepatocytes to respond to the potential cholestatic effects of a compound in a relatively short time period, in this case being 24 hours. So it integrates effects on all of these pathways. So if we look in a little more depth at the two types of media and some compounds for this that we typically use as controls. So cyclosporine here, as you know, is a very potent BSEPT inhibitor, but if the cell can respond and adapt to that, you won't see any toxicity in this sensitized culture media because the cell can adapt. And you also don't have direct toxicity for cyclosporine at the concentration that was used. With other compounds, such as imatinib, you see a significant direct toxicity component, and you also see some increase in the sensitization media. So this is indicating there's direct toxicity in addition to some other potential hepatotoxicity going on. For triglyceride, that in the standard media is not toxic, and I'll show examples of that where we've done dose ranging studies with that. We can go up very high in concentration and not see that. But in the sensitization media, you do see toxicity with triglyceride very consistently. In fact, so consistently that this is our positive control for that. So other things that we had to consider in the assay of these 50 compounds is what concentration do we use? And typically there's with these compounds, there's information on either the systemic Cmax or steady state concentration. We've known from previous work that we've done that for orally administered drugs, the portal vein concentrations are typically higher than the Cmax, and the range for that is somewhere within 10 to 20x the Cmax. And so that's what these compounds were evaluated at, both for triglyceridone, so at about 100 micromolar, which is about 10, 15x the Cmax, we're seeing some effect. Cyclosporine we've evaluated, again, much higher, and we've not seen any effect with that for cyclosporine. So cyclosporine is a potent BSEPT inhibitor, but again, the hepatocyte can adapt to that BSEPT inhibition. And triglyceridone, it's important to note triglyceridone and its metabolites, primarily the sulfate metabolite, is a very potent BSEPT inhibitor, but also triglyceridone is a relatively weak FXR antagonist, but it does have FXR antagonism properties. And so we only see toxicity for compounds that impact multiple pathways, and this can either be inhibition of basolateral efflux in addition to BSEPT inhibition, or FXR antagonism, which prevents the hepatocyte from responding to these increased intracellular concentrations of bile acids. And as I mentioned earlier, we chose an LDH readout to try and link it to the clinic more effectively. So the compounds I'm going to show you were selected from work published by both Morgan and Dawson, and they were all based on IC50s against human BSEPT in a vesicle system. For these drugs, since the portal vein we wanted to try and take into account higher portal vein concentrations, and so the test concentrations that we used were generally in the 10 to 20x the systemic Cmax, or for some of them we were limited by solubility. Also, the NIH liver tox database was used to identify compounds with clinical hepatotoxicity potential. The liver tox database is a tool out there that's available to everyone. However, they do have some issues with how they classify DILI compounds with that, and as you really have to spend quite a bit of time in that database, going back to some of the original reports or references to really determine which compounds and the incidence with which they cause drug-induced liver injury. And so we've spent a lot of time on that. So here's the list of compounds that we've looked at in this initial assay. As I mentioned, these were all from the liver tox database. The concentrations that we used were 10 to 20x the systemic Cmax, or in some cases a steady state concentration, or if we were limited by solubility. We've identified, based on our assay, some false positives, some false negatives, and a number of true positives for this, and have linked those to drug-induced liver injury for this. And so you notice there are some where there's a lot of idiosyncratic reactions that probably this or any other assay may be very difficult to capture for that. So when we look at all these compounds, and typically we've screened those for this assay, both in the standard media and in the sensitization media. Again, the sensitization media just creates the conditions so that we can see and get a signal in a short 24-hour window for this. And you can see there are a number of compounds that have a very low potential for hepatotoxicity. There's some that kind of are intermediate or relatively weak, and there's some that are very, very high, and there's some also where we see toxicity in the standard media, which is indicative of direct hepatotoxicity for this. We can take these some of these hits for this and do more of a concentration range for this, and we can actually estimate, what a no effect concentration would be for this. And if you look at if you look at this, the toxicity is only observed and for these compounds when they impact multiple pathways, and that includes inhibition of BSEP and or basolateral efflux transporters or our FXR antagonists for this. But you can definitely, see with triglyceridone, you can see a dose response for deferosox also, and you can generate concentrations where you don't see any response from that. Likewise for ketoconazole also. And in addition, we can look at, do further dose ranging concentrations. These are just some additional compounds for this, but you can see previously, we didn't see a signal for lapatinib, which does have some liver tox issues, but if you go higher in concentration, you can now see a signal for that. We can see dose responses for many of these compounds, deferosox, as I mentioned before, amiodarone, ketoconazole, fluvastatin, for example. So you can use the values that you see for concentrations here then to relate that to potential clinical exposures and get an idea of the potential of your compound to cause cholestatic hepatotoxicity. So we've done additional work again looking at, I've just highlighted a few compounds here, some of the glitazones and some of the statins. Again, cyclosporine is our negative control for pioglitazone, rosiglitazone, simvastatin, all of which are clean clinically. For cholestatic hepatotoxicity, we see no signal for that. Again, for ketoconazole, triglyceridone, and deferosox, we see signals for those, but only in the sensitized culture media, indicating that it's not a direct toxicity mechanism that we're picking up here. In addition, we can also do more extensive dose ranging studies. So here I'm showing a study where we went over a fairly large concentration range, 1 micromolar to 100 micromolar for triglyceridone, pioglitazone, and rosiglitazone. This was under the standard conditions, a 24-hour exposure with either the standard media or the sensitization media. We measured cytotoxicity by LDH. We also looked at ATP depletion. I haven't shown that here, but we also generate that type of data. The one important thing to notice is that under the standard media conditions, we don't see any signal for that. So these concentrations are not in a range where they cause direct toxicity for that. And actually, it's only under the sensitization media, and the only compound, triglyceridone, was the only one that generated a signal for that and actually increased the LDH secretion, We didn't see anything with pioglitazone or rosiglitazone under these conditions. Also, again, our negative control cyclosporine, we did not see any changes for that compared to DMSO control for that. So it's a very, very nice showing that you can actually differentiate the cholestatic hepatotoxicity of triglytazone from two other similar analogs, the rosiglitazone and pioglitazone, for this. We've also done some looking at the variability for this, again, primarily using our negative control cyclosporine and the positive control triglytazone. If we look at interplate or between plate with different donors, we'd see a very consistent response with cyclosporine and also a very consistent response with triglytazone with fairly low interplate and intraplate variability for this. So I think it's a fairly sensitive assay. If we bring this back to the same compounds that I showed before, where we're looking at the database from Morgan and Ryan, we see that if you use BSEP prediction, if you use the IC50 against human BSEP prediction alone, you get a lot of things wrong. But if you add an assay that integrates all of these factors, you have a much better potential to predict the cholestatic potential of your compound. And if we do a contingency table analysis for this, much like we did before, you can see we have a very good sensitivity and specificity for this with a fairly good accuracy of over 90%. So we're very satisfied with this, and we're looking forward. We're running additional compounds in this to make sure that these incidents hold up and continues to be predictive. So just some final thoughts on how you could use this assay in drug development. And we kind of see three areas for this. One, earlier in mid or late discovery to identify structure-activity relationships for cholestasis and to actually screen and identify compounds that have the potential to cause cholestatic hepatotoxicity. And then if you have a little bit later in development, if you want to perhaps understand the mechanism of this, if you get a hit, you can use this to understand, are you inhibiting BSEP? Are you inhibiting basolateral efflux? Or does your compound have FXR antagonistic properties? And then a little bit later in development, you can use this to predict clinical hepatotoxicity, but also to evaluate the drug interaction potential if you have multiple pathways impacted. So we could envision, you may have a compound that inhibits BSEP that would not necessarily cause an effect like cyclosporine, for example. But if it's administered with a compound that has FXR antagonistic properties, then that interaction could cause a problem with that. And when you look at the liver tox database for a number of these compounds that they indicate have liver tox potential, these patients are on or have received multiple drugs for that. So a lot of times it's not just a simple, single drug administration. So thinking about the drug interaction potential for this may also be very important. And the nice thing about this is no matter where you use this in drug development, you're integrating multiple mechanisms for this. You have in vivo relevant intracellular concentrations, and you can use the same model at different stages to answer all of these different questions. So the data is translatable for this. So to summarize, this C-DILI assay to evaluate cholestatic hepatotoxicity uses human hepatocytes. It has a fairly high density format, 96 well, uses a short experimental time, 24 hours, that integrates many factors, and it has a very simple LDH readout for this. It integrates effects, acute effects of the compound, so it takes into account metabolism. It also takes into account uptake and efflux transporter function, and then integrates that with chronic effects of the compound. So this whole concept of adaptive response by the liver on hepatobiliary disposition of bile acids. And this involves regulation of various transporters and metabolic enzymes for this. And it also integrates effects on the synthesis of endogenous bile acids. So taking all of these together, it's really the net effect of all of these processes on the bile acid disposition that determine the cholestatic drug-induced liver injury potential of a compound. And so I'd like to end there and take any questions, and just an acknowledgement of the people that have done a lot of the work on this. I want to thank Jonathan Jackson, who did a lot of the regulation work with this, and our analytical people and other people in the lab. If you have any additional questions or need any additional information, please feel free to contact me or any of our business development associates, and their contact information is listed here. And I thank you for your time and attention, and would be happy to answer any questions.
Okay, thank you. And the floor is now open, so if you have a question, please type it in the question box, and I will read it out, and we'll get an answer. And like I said earlier, if you tuned in later, the handouts will be emailed to you as soon as I get them, so they just will be sent to your email address. Are there any questions? Okay, going once. Going twice. Okay, well, it looks like there aren't any questions. So with that, we will wrap things up. And once again, thank you, Dr. Brower, and thank you those for attending. And we hope you found it useful.
Thank you.